Thank you, Christina, and welcome everyone. The Committee on Credible Practice of Modeling and Simulation in Healthcare welcomes you to this webinar. My name is Joy Koo, and I'm one of the executive committee members on the committee, and I'll be serving as the moderator for the webinar today. Very pleased to welcome today's presenter, Christina Friedrich from Rosa & Company. Next slide, please. So the Committee on Credible Practice of Modeling and Simulation in Healthcare, or CPMS, is an initiative started by a consortium of U.S. funding agencies called the Interagency Modeling and Analysis Group, or IMAG, as you see listed here, and the Multiscale Modeling Consortium. Our goal is to establish guidelines for simulation-based medicine that's to increase the adoption of modeling and simulations within clinics and healthcare in general. Next slide, please. The key goal of our webinars is to, one, to showcase how individuals have translated their modeling and simulation research into clinical use, and two, to engage the community in discussions on best practices for modeling and simulation. Next slide, please. So before we get started, I just want to share a few reminders about the webinar format. Questions will be addressed at the end of the presentation using the Q&A panel. This is all text-based. And if you need more help, please consult the guide that's available on the events page. The URL is listed there. Next slide, please. And now I'd like to introduce Christina Friedrich. Dr. Friedrich is the Chief Engineer at Rosa & Company with over 17 years of experience in applying mathematical models of biological systems to support development of biopharmaceuticals and consumer products. Her therapeutic area experience includes diabetes, blood disorders, CNS disorders, oncology, respiratory diseases, skin diseases, and other immune system dysfunctions and inflammatory processes. While at Rosa, Dr. Friedrich developed and published the first peer-reviewed journal article on the qualification of mechanistic mathematical QSP models. Prior to joining Rosa, Dr. Friedrich was the Director of Core Product Development at Intelos Incorporated, the first-generation leader in physio-PD-style models, where she spearheaded development of modeling methodologies and contributed to foundational patents, including on the use of virtual patients and populations to explore biological variability and uncertainty, and on the identification of biomarkers. Dr. Friedrich completed her BS at MIT and received a PhD in Management Science and Engineering at Stanford University. We're very pleased to have her here today to discuss with us the qualification process and method that she's developed at Rosa. So thank you, Dr. Friedrichs.
Okay, thank you so much, Joy, for the kind introduction and the invitation to speak here today. So as Joy mentioned, I will talk about how to ensure QSP model quality, and I will take you through the model qualification method that we have developed here at Rosa. So first, I'd like to start with a very brief reminder of what QSP is, and this goes back to a white paper that I'm sure many people on this call are very familiar with from 2011. And I'd just like to call your attention to one aspect of the proposed working definition for QSP that's highlighted here, where it states that QSP will accelerate drug discovery and development by helping to identify and validate targets and druggable networks, uncover drug response biomarkers, design better drugs and drug combinations, select appropriate doses and dosage regimens, and identify those patients most likely to respond to new therapeutic agents and combinations. And the reason I wanted to highlight that is just to point out that that is a lot of different applications for this new modeling technique called QSP. So how can QSP really do all that? At Rosa, we like to think about it this way. QSP helps primarily by reducing risk in drug development by improving understanding of how drug activity influences clinical outcomes. So the central question in drug development is really, we have some preclinical evidence, perhaps a compound is inhibiting a certain pathway that you think is involved in the disease pathophysiology. So there's some mechanistic hypothesis that leads you to conclude or to develop a drug to hit that particular pathway under the assumption that it may benefit patients by improving clinical outcomes. And the central question in drug discovery and development is always, will this perturbation that we can achieve with our compound really have the intended effect on the pathway, and will the pathway change, in fact, then lead to an improvement in clinical outcome? So QSP helps in this process by really formalizing, quantifying, and making testable much of the scientific underpinnings that go into making this connection from mechanism to clinical outcome. So in other words, QSP is really taking the science and making it more formal and testable, and contextualizing the preclinical evidence that you have. That makes it a little bit different from other modeling approaches that are used during drug development. So traditional modeling techniques use specific types of data generally generated during development for a particular compound to inform the next step or the next decision in development. QSP has a much broader set of applications and is often used to extrapolate out much farther. And I should just mention that PhysioPD is the term that we use at ROSA for our brand of QSP, mechanistic QSP modeling. So how can QSP make these much farther extrapolations? It's really by drawing on a lot more data. So in addition to considering any data that are generated in the course of development for a particular compound, we also draw on a variety of public and other proprietary data that help inform that mechanistic component of the representation. So data about the disease, data about how other compounds may be targeting different pathways to help produce a clinical improvement, et cetera. That's all considered when we put a QSP model together. And because we draw on a much broader range of data, we can then support farther extrapolation. So for example, we may do a project where we prioritize targets. And you may not even have compounds yet, but we're just thinking about targets, and we are predicting their potential clinical efficacy given everything else we know. So, and of course, the challenge of prioritizing targets is one that we face anyway. The QSP modeling just helps make better use of the existing data that may help inform that particular decision. So again, QSP is a way to do science. So from that description, hopefully it's fairly obvious that prescriptive validation techniques that assume specific data and a specific purpose for the model really are neither necessary nor sufficient for qualifying these types of models. Rather, QSP's broad range of different applications and use of different types of data really calls for a flexible qualification framework that addresses key aspects of model quality. So the question we're trying to answer is, are we doing the best possible science when we're putting the model together? So I'd like to take you now through a few of the considerations that we think about when we think about building trust and confidence that the model is capturing the science appropriately. So the first area of model quality that we always think about is relevance. Are you building the right model for the question that you're asking? And before you can even ask that question, you have to make sure that the research context is very clear and that everybody's on the same page with respect to what you're trying to address with the model. And then we want to set functional and biological scope according to the research context. One very easy mistake to make when doing these types of building these types of mechanistic models is to include too much detail on mechanisms that may not be directly relevant to the question that you're asking. We also want to make sure that the representation of biology and pharmacology reflects the current state of knowledge. And by that we mean we want to be able to look at the diagram that we're putting together that graphically represents the model and show that to scientists in the field and have them say, yes, that this is indeed how we think the biology works. So we get a lot of confidence from being able to do that type of qualitative validation with experts in the field. Secondly, uncertainty. There's always uncertainty about the biology, and that becomes particularly relevant when you're trying to build a mechanistic model of how the biology gives rise to the clinical outcomes that we see. So modeling does not create uncertainty, and it also doesn't make uncertainty go away, but it does allow you to be much more systematic and disciplined about how you deal with that uncertainty. So the questions that are relevant there are, given the biological uncertainty, how robust are model results and conclusions? Would your recommendations actually change under different assumptions? And which uncertainties of the many that may be out there really pose a risk to the program? And what experiments could potentially resolve those uncertainties so that you can move forward with more confidence? Again, it's all about de-risking the development process. We also worry about variability, and your modeling project should address variability. We particularly care about mechanistic variability. How does mechanistic variability connect to the outcome variability? So how do known differences between patients affect clinical outcomes? And what biological mechanisms may explain the range of observed clinical or preclinical outcomes that we see? Again, it's the modeling that we do in QSP is not about describing the variability in a particular data set precisely, but rather about formulating and testing hypotheses about the mechanisms that may be leading different patients to respond differently. And finally, of course, we need our model to be in good agreement with data. So here we think about matching relevant data, both at the clinical or whole system level, as well as at the subsystem or mechanistic level. And very importantly, we need one single representation or parameterization of the model to be in agreement with all of the data that we can find, subsystem level data as well as system level data. So the subsystem level behaviors need to give rise to the system level behaviors. And also, of course, we want the model responses to a variety of different tests to be consistent with current knowledge and expectations. Again, we don't focus so much on matching a particular data set, but rather on perturbing the model in different ways so that we can test that the response of the model to these different types of perturbations is still reasonable and consistent with what's expected. So we do have a publication for anyone who's interested in reading more about this that goes into a bit more detail on each of these criteria for relevance, uncertainty, variability, and matching data. I won't go through that in more detail right now, just in the interest of time. But I did want to mention 2 more points here. So the first is that in our practice, we use this model qualification method for project management and indeed documentation throughout the project. So at the very beginning of the project, we make sure that we discuss with the client each one of the criteria in the model qualification method, and we start to talk about our plan for ensuring that the model meets the necessary qualification criteria in each of those categories. And then as the modeling progresses, we build a very substantial MQM document that documents all of the various considerations that went into building the model, representing the biology in a certain way, documents uncertainties, etc. So we begin with the end in mind by stipulating a priori what criteria does the model need to meet when it's done. I'd also like to point out that this is a general framework and it can be customized, and in fact should be customized, and different types of qualification techniques will be relevant for each project depending on the research context. And research context here is defined as the research questions that you're asking, as well as the data that you are using to build the model. So that's the general framework. What I'd like to do next is to take you through a couple of case studies from the ROSA practice that illustrate how the MQM can be used to guide these types of projects. So the first case study is in chronic obstructive pulmonary disease, or COPD. I can't disclose the client in this case, but I can tell you that they came to us with a need to clarify the mechanism of action of a novel compound and anticipate the clinical response. So the client was developing a novel compound as a second-line therapy for COPD. They had preclinical data from a in vivo model that suggested an efficacy and safety advantage compared to roflumilast, which is a PDE4 inhibitor that has a limited therapeutic window due to safety signals. The mechanism of action of the novel compound was actually not fully elucidated. The client had some hypotheses and some data, but there were still some questions around exactly what the compound was doing. And it was also not clear how preclinical results would translate to human patients. So what were the research questions in this case? First, does the current understanding of the mechanism of action actually explain the compound's efficacy? And is the mechanism likely to translate from preclinical to human? So the data that we had in this case, there were no clinical data for this compound. We had preclinical data for that compound in the in vivo model, as I mentioned. We also had comparator data in that in vivo model. Additionally, we had public data on both preclinical and clinical data for roflumilast and other relevant test therapies. And we also had, of course, the broader set of literature describing the mechanisms of COPD pathophysiology, as well as the role of the compound's targets in the pathophysiology. And that's important because when we represent a compound in a mechanistic model, what we're really representing is the mechanism of action. Where does the compound hit the biology? And then everything downstream of that is just a function of the mechanistic representation that we've put together. So we really expect the model to reproduce the effects of different compounds. So let's take a look at how we qualified, what the qualification plan was for this research project. So on the relevance front, we wanted to represent just sufficient biological scope and detail to represent the compound's mechanisms of action and address the research questions. We, of course, wanted to use the current understanding of COPD pathophysiology to inform the representation. And we also wanted to use biological subsystem behavior data to inform subsystem calibration. In terms of uncertainty, we wanted to first of all focus on uncertainty relevant to the novel compound. And that included some possible measurement issues with some of their preclinical data. They had some somewhat conflicting data, and they were concerned that some of the work may not have been as solid as it should have been, and they wanted to know what their risk was with moving forward with under the assumption that the data were correct. And then, of course, as I mentioned, they also had uncertainty about what the compound was actually doing. In terms of variability for this particular project, we did not focus on any evaluation of clinical variability because of where this compound is in its development. That wasn't necessary at this point. We did take a look at some species differences that might be helpful in the preclinical to human translation. And in terms of data that were available, we, of course, wanted to use all the mechanistic data, so subsystem and pathway level data, to build the mechanistic representation, and we wanted to test that that representation could produce the system level behaviors that we're expecting, both in the untreated and the treated case. We used compound-specific data for the compound implementation. We expected that the model should exhibit a typical roflumilast response. So again, right now for this project, we were not concerned about the range of responses. We were just focusing on a typical response. And we also wanted response to long-acting beta agonists and inhaled corticosteroids to be typical. So these are our additional test therapies that we use to check that the model was giving reasonable responses. So let's take a look at the model that we built. This is a high-level overview of the COPD model, and I don't have time to go through this in a lot of detail. I'd just like to mention that it does include what I would call various subsystems that are relevant for COPD, including immune cell recruitment and activation, epithelial cell activation, production of various mediators, alveolar wall destruction, goblet cells and mucus production, airway smooth muscle activation and contraction, and fibrosis. And it also included quite a bit of detail on cyclic AMP dynamics, and that's a direct result of the fact that we are using roflumilast as a key comparator therapy here. And that's, so the cyclic AMP production is relevant for the roflumilast mechanism of action. So I'd just like to point out that this diagram really is very much reflective of the research context that we were facing here. We put in detail where we needed it, such as for cyclic AMP, but other parts of the system are modeled at a much higher level. So, for example, goblet cells and mucus production, that's this section down here, that's modeled at a pretty high level and doesn't include a lot of detail. You can imagine that if the target of their compound were had something to do with mucus production or were located on the goblet cells, that would be something that we would potentially build out in a lot more detail. So this diagram really reflects that relevance criteria that I talked about earlier, where the detail was only included where it was needed, but we had enough detail overall to be able to reproduce the clinical level responses based on the mechanisms of the different compounds. So interestingly, something that we found out when we implemented the novel compound based on its hypothesized mechanism of action, if we compare on the left response to roflumilast in terms of immune cell infiltration into the lung tissue versus the response to the novel compound, we see easily that the roflumilast does a much better job, actually, of reducing the immune cell count in the tissue. The novel compound is having a pretty minimal effect. So this simulated efficacy is actually well below what was expected based on the preclinical results. And we then did some additional research that demonstrated that that unexpected result actually could not be explained by species differences or by any of the methodological variability in the in vitro compound characterization that I mentioned earlier. So as you'll recall, one of the uncertainties that we wanted to focus on was this uncertainty around the mechanism of action. So what we showed was, first of all, that the favored hypothesis of what the compound was doing was not actually sufficient to explain the preclinical efficacy. We therefore then explored some additional mechanisms of action. So we were able to add these hypothetical additional effects to the platform and then rerun the simulations. And I'll just mention that what the simulations then showed is that the additional mechanism of action effects could actually be synergistic, the two that were hypothesized. And while either one alone was not enough to explain the efficacy, the combined effect of both of those mechanisms would in fact get the compound much closer to what would be expected based on the preclinical results. So the bottom line from this project was that the mechanistic modeling really facilitated a careful, systematic, and quantitative evaluation of the evidence and helped the client avoid possible risks of going into clinical development with an incomplete understanding of the mechanism of action. We also were able to support the scientific rationale for adjusting the development plan by really spelling out which parts of the biology we relied on to make come to the conclusions that I mentioned. So this was not exactly what the client expected when they engaged us, but it was extremely helpful for them to understand better what their asset was actually doing and help them create some additional mechanism of action hypotheses, which then, of course, could be tested in the model that we built. So that was the first case study. The next case study is in cardiovascular disease. And in this case, I can disclose that this was done with Sanofi colleagues. And we've actually presented this work both at ACOP as a poster and then in a publication. So I'd just like to refer you to this journal article for additional details about this work. I'd also like to mention that the figures that I'll be showing for the rest of this case study actually come from the publication. So again, you can consult that for more information. So Sanofi was interested in better understanding the variability that's seen in low-density lipoprotein cholesterol response to different drugs. And they also wanted to begin to investigate how LDL lowering is actually mechanistically connected to plaque dynamics. So a little bit of background here. Reduction of LDL cholesterol is associated with lower morbidity and mortality from cardiovascular disease. And PCSK9 inhibitors, such as Sanofi's antibody, alirocumab, significantly reduce LDL cholesterol. The impact on cardiovascular disease outcomes is still under clinical investigation. And I would say in general in this field, there's a great interest in better understanding the mechanistic connections between the lipoprotein effects and the cardiovascular outcome effects. So the client's research questions in this case were, what mechanisms may underlie the range of LDL cholesterol responses under different therapies? And what is the potential impact on atherosclerotic plaque dynamics? The data that we had included some clinical data for anti-PCSK9 and indeed alirocumab specifically. And we also had data for other LDL-C lowering drugs that showed, again, a range of responses for different patients. We also had biologically informed hypotheses for mechanistic drivers of clinical variability. These, of course, were based on literature as well as some additional data that Sanofi brought to the table. And then we had, as always, the public data, which include mechanistic data for lipid metabolism, as well as mechanistic data for plaque formation, role of inflammation, interaction with lipoproteins, etc. So in this case, the qualification approach accounted for the fact that there's much more of an emphasis here on variability. So I'll just briefly mention that in terms of the relevance criteria and the data criteria, those are quite similar to the last case study. So again, we want to represent just sufficient biological scope to address the questions and implement the compounds mechanistically. And we used systems level and subsystems level data and knowledge to inform the modeling. And then we tested the model against those various pieces of data and made sure that the subsystem level calibration gave rise to the right clinical level behaviors. The additional test therapies that were included were statin, ezetimibe, fibrates, and of course the anti-PCSK9. And we wanted the model to fall into the expected range of responses for all VPs. But the real meat of this project, as I mentioned, was around variability. So here we were interested in evaluating the effects of mechanistic differences on the response to alirocumab and other therapies. And the way we actually evaluated this was by creating virtual patients, which are essentially different versions of the model that had known or hypothesized mechanistic differences that could lead to the differential responses to therapies. One question that we often ask in this type of project is, how different can those virtual patients be from each other and still satisfy all of the data constraints? So we're really kind of testing the corner cases first to try to see what the most extreme types of patients may look like. The uncertainty exploration in this case was limited to documenting relevant biological uncertainties for future exploration. And we also did make use of the fact that there is biological uncertainty to set some of the parameters for the virtual patients that we created. So having an uncertainty gives us a little bit of leeway to change the virtual patients to try to get them to behave differently. So this project is really all about hypothesis exploration around the question of what differences in pathophysiology give rise to differential responses to cholesterol-lowering drugs. So again, let me take you briefly through the diagram that was that represents the model that was constructed. Not to go into any detail, but the model included quite a bit of detail on hepatic, peripheral, and plaque lipid metabolism, and then also some additional plaque dynamic equations. Again, more detail on this is available in the publication. These figures, again, just show some of the subsystem and whole system level testing that was done of the platform. Don't have time to go into detail on that, but suffice to say that all of this is documented, of course, in the MQM document. And those documents typically for a model of this scale would run to about 150 pages or so, really describing all the thinking that went into the model and as well as the testing. So as I mentioned, the real meat of this case study was in the exploration of variability. So we started that exploration by creating 4 different virtual patients to represent different types of real patients. To facilitate comparison, we wanted those 4 additional patients to have a similar clinical presentation on background statin therapy, so that when we started them on the additional therapy, they would all start at the same place. We created the patients by altering some key parameters that were expected to have an impact on the response to the therapies, as I mentioned. So some examples there include some of the parameters governing untreated hepatic cholesterol synthesis, LDL receptor turnover, PCSK9 turnover, etc. In all cases, the parameter values that were chosen were still within the reported data ranges. There are some details about this available in the publication. So the VPs were deliberately designed to explore variability, in this case in response to statins and alirocumab, where VP1 and 2 were strong and weak statin responders, and VP3 and 4 were strong and weak alirocumab responders, respectively. So the process of defining these virtual patients was actually very interesting in and of itself, because it amounted to an exercise in testing different hypotheses of what kinds of changes in the mechanistic pathways would lead to outcome level variability of the range that we were looking for. And this figure here just summarizes the different behaviors of the 4 VPs, the 4 additional VPs that were created to alirocumab alone and alirocumab with atorvastatin on the right. And you can see indeed that, for example, VP4, which was designed to be a weak responder to alirocumab, does indeed have the least response to these treatments. So this certainly does not prove that every patient who's a weak responder to alirocumab has the same characteristics as VP4, but it does confirm that, first of all, a range of responses is possible under different hypotheses about the pathophysiology, and that the particular assumptions that we made for VP4 are consistent with a low responder. So it really gives a lot more mechanistic insight into what's happening in response to these different therapies. These first VPs are really just a starting point. Certainly, many more VPs can be created to investigate different hypotheses and gain additional insight into how the mechanisms give rise to the outcomes. I didn't really have time to talk about any plaque outcomes here, but the hope, of course, is that in the future we can gain some very interesting insights about how LDL cholesterol-lowering therapies impact the plaque dynamics, and that in turn could be very helpful for predicting cardiovascular risk. So again, we showed that we are able to recapitulate the effect of cholesterol-lowering therapies on lipid profiles, as well as plaque size and plaque composition and stability. And by using different virtual patients, we were able to investigate plausible hypotheses of mechanistic variability that may be giving rise to the outcome variability. So hopefully you can see that this type of platform can then be used, for example, to inform clinical trial designs that would work for different types of patients, or perhaps to define biomarkers of response or inclusion criteria, or to really just support a more personalized medicine approach. So hopefully I've been able to give you a sense of the reason why qualification for these types of models is really not a one-size-fits-all proposition. There's a wide range of data and different types of applications for these models, and so prescriptive validation approaches really are not applicable. Rather, model qualification should be appropriate for the research context, so the questions being asked and the data available. And in our practice, we found that this model qualification method provides a general, customizable qualification framework for these types of models, and that by beginning every project by talking about how are we going to build confidence in this model and illustrate that it's fit for purpose, that's been extremely helpful in reassuring the client and building trust in the representation of the biology. So the last thing I wanted to mention here is that I'm sure many on this call are familiar with the CPMS 10 not-so-simple rules for credible practice of modeling and simulation in healthcare. And when Joy and I first started talking, we immediately observed that there was actually quite a bit of overlap with the criteria in the MQM. So the 10 rules are listed out here. They include defining the context, using appropriate data, evaluating within the defined context, listing limitations, version control, documenting, disseminating the model, getting independent reviews, testing competing implementations, and conforming to standards. And I won't go through a detailed analysis of this, but in looking at that, I think it was immediately obvious that especially the relevance criteria maps onto many of these rules. And I think some of the other MQM criteria also fit quite nicely within some of these rules. And I also wanted to mention that while not necessarily spelled out in the MQM, it certainly is standard ROSA practice to follow some of these other rules, such as version control, disseminating, so we share the model with our clients, and certainly conforming to standards. So I think there's actually quite a bit of overlap, and it's quite interesting that through different paths, we've kind of converged on many of the same criteria for quality in these models. So that concludes my presentation, and I'll turn it back over to Joy now to manage the Q&A.
Thank you so much, Dr. Friedrichs. That was a great talk, really insightful in terms of both the qualification method as well as its application. So I want to open the floor now for your questions. All of the questions are going to be text-based, so if you want to ask a question, go to your Q&A box, should be on the bottom right of your screen. Type in your question and make sure you select to ask all panelists so that we actually see your questions and can address them. So I'm going to go ahead and, while we're waiting for people to type in their questions, I wanted to ask you a question about the documentation piece of it, because it sounds like you guys internally and what you share with your client is a very detailed, very rich set of documentation. But then when you and your Sanofi colleagues went to publish it, I'm not sure how many pages your publication was, but I'm sure it was much smaller than what you had internally. And I wondered if you had thoughts about how you converted all that information and condensed it down to the publication. Should there be other mechanisms for also sharing the other information to increase kind of confidence in the model?
Yeah, that is a great question, and it really points to the complexity of these models. I mean, I think what I showed you gives you a pretty good idea of the fact that you know, there are a lot of biological consideration, evaluation of data, discussion with experts, et cetera, had to go into defining, you know, every single connection in that model. So yes, there's a lot more that one would need to know in order to certainly use the model and even to interpret results appropriately. So on one hand, yes, I agree that having a mechanism for sharing more of those considerations would definitely be helpful. maybe now that I'm thinking about it, there may be sort of two separate sets of users for this type of information. I mean, not everybody who sees the model has to be able to reproduce the to use the model to generate new insights, et cetera. So part of why we are so detailed in our documentation is because we want to empower our client to be able to continue to use the platform. So in that respect, that's one reason why we do go to a lot of detail in our documentation. I don't know how much would be reasonable to put out in the public domain, but I don't, if someone is really interested in all the details, certainly I don't see any reason why one wouldn't just go ahead and publish all of that information. So yeah, I think to really understand the model, you need every last bit of the information that's in that MQM. But yes, you're right, the publications are limited to really just sort of the top line data that went into it. That's also one reason why having this MQM framework is helpful, because even if we can't list out every single activity that we undertook, hopefully it's helpful to at least understand that we take all of those aspects of model quality seriously and did address them.
Great. Yeah, I think it's a complex question. It's interesting to get your insights onto that. We have a question from Amit Erdemer from Cleveland Clinic. He says, can you elaborate further on how you get client buy-in to conduct model qualification? He says, I suspect some may have a sticker shock and the activity should fit in the resource and time constraints.
Yes, that's a really great question. So the QSP modeling, as I said, is best suited to address questions that really require extrapolation beyond sort of the next step by drawing on mechanistic insights. So if you think about it, these models are, they pull together a lot of science, and yes, that does take time. It also then, though, allows you to continue to inform additional questions that may come up for that in that therapeutic area, both for the compound you're currently considering and any follow-ons potentially. So yes, you have to set expectations appropriately for what it will take to put these models together. And what we find is that we do have a lot of repeat business. The initial convincing may take some time, but clients do come back because they see the value of the approach. And our philosophy is always to be as transparent as possible, to involve the client at every step of the way so that they really feel ownership over the model, and they understand the judgment calls and decisions that were made to put the model together in the way that it was. So in terms of ensuring that they, in terms of gaining trust, that's really a process that doesn't just happen sort of at the end in a qualification step. It really happens along the way in those regular meetings with the client and the full transparency. And as I mentioned, we also transmit all of the modeling files as well as all the documentation on a regular basis so that our clients can follow along as we're putting the model together. So I hope that addresses that question to some extent. Yeah.
Thank you. So along those lines, I think we had talked earlier also about, for instance, you mentioned you have a lot of repeat clients and projects. And so that first step, though, of how do you get someone to trust your model and to believe that you're going to be able to, your model is going to give them the answers that they are seeking, I think that's the question that relates for your first-time customers, but also for researchers who are developing their models, from scratch. Could you address some of the, I guess, process that you think is helpful in that regard?
Yes, absolutely. I didn't mention this before, and maybe I should have, but the ROSA modeling teams are always comprised of experts in modeling who typically have an engineering background and experts in the science who have a life science background in the area that we're modeling. So it is very powerful for our clients to be able to talk science. We're not really talking about the model most of the time. We're talking about, here's what the science say. We're proposing that for the purpose of modeling, we're going to interpret it in this manner, and here are the simulated model results that are consistent with that interpretation. So we do that dozens of times over the course of developing a model. And I find that being able to talk science with the client team, which, by the way, also should be a cross-functional team. We don't just want to be speaking with the modelers. We want to speak with people, representatives of different functions, including the therapeutic area experts, so that we can really discuss, again, how does this mechanistic model capture the science, how are we interpreting different data, et cetera. And I think that conversation really builds a lot of confidence that, again, the model is representing the biology in a way that is appropriate. So that's one way. And, like I said, just being as transparent as possible, capturing relevant assumptions, being explicit about uncertainty. I think all of that helps. Having said all that, it's much easier to explain that once a client has gone through the process. So it is still difficult to set the right expectation for how the modeling process, the process of going through the modeling with us is, I think, going to instill confidence. But it's always a bit challenging to communicate that before we've actually done the project. So that's definitely still a challenge in this field in general, I think.
Right, and so along those lines, Lala Mulugete has the question, when you're vetting and validating the conceptual model, how do you go about selecting that subject matter expert? And how do you do so to ensure there's no conflict of interest?
Yes, okay, that's a good question. I may need to get a little more information on the conflict of interest concern, but in general, as I said, ROSA brings disease area and biological expertise to the table to help us, make sure that we're interpreting data appropriately. It is not uncommon for us to be somewhat more objective in interpreting literature data than maybe what the client has considered as possible interpretations of what's going on. So those are useful discussions to have. It's always backed up by the science. I will say that also, though, we want to reflect the client's understanding of the biology. And there is no one right answer necessarily of what is actually happening in the science. At least there may not be sufficient evidence yet to definitively say what's actually going on. But the client's disease area experts' opinions are informed by years and years of research in the area. So if there are no definitive data to suggest that their understanding is not appropriate, we certainly would, for example, accept a client's preference on which hypothesis to go with, and then just document that in the document, in the MQM document. Our goal is to help our client make a decision, and modeling or no modeling, they would certainly rely on their experts to help inform that decision. So I think it's actually an important point that the model that we build for one client may not be exactly the same as the model we would build for another client, because it's possible that their experts have come to different conclusions, and that's biology, and that's certainly a reality. So for our goal is to help our client make a decision that they're comfortable with and that they're confident about. So I hope that sheds some light on the question, but feel free to ask a follow-up if that didn't quite address what you're asking.
So I think it addresses it sounds like you guys have a subject matter expert it's an in-house expert. So it's not so much
Yes.
that you go out and look for a consultant who knows about COPD. You have someone in-house who knows about COPD. So
yes,
that's
right. So, well, it's a combination, actually. We do at times also go out, and we actually have a set of consultants that we've worked with in the past that we could engage for specific questions about a disease area that maybe we don't have as much in-house expertise on. But we also, there's a specific skill set for a scientist to support modeling, and we find that our in-house modelers, our in-house scientists are very good at finding the types of data that are most useful in building these types of mechanistic models. But yes, we do at times also consult other experts in the field.
So along those lines, Lalam had the follow-up clarification in terms of the conflict of interest. So when you go and select those consultants, how do you ensure that they don't have a bias, for example, a biased interpretation of the proposed modeling and simulation approach? So for instance, they might have some financial or intellectual interest in pushing one particular model approach. How do you ensure that they're coming with a kind of a comprehensive look at what's out there and what's really best to use?
It's not something that we've had to deal with very much, I have to say. I think our best option is to really form our own opinion, like I said, based on the independent research that we do of the literature. I think it's fair to say that there are certain people who tend, scientists who maybe are known for having strong opinions for one or another hypothesis, and we don't tend to try to work with people who are known to be biased. So I haven't really encountered that particular scenario very much. Has not really been a big issue for us.
Okay, great. So another question from Ken Wilkins, who says, interesting talk. He wants to know how you handle metadata or data or unpublished findings that a client considers proprietary or potential IP.
Oh, okay. Yes. So I didn't mention this, but every model that we build is built specifically for the client that we're working with. And those components of the model that are informed by proprietary data are completely firewalled and will never be used again in another context. So if part of the question is around IP, we have pretty simple IP arrangements that make it very clear that the client continues to own any IP, and none of their IP will be in any public or reused model unless they choose to move ahead with publishing the model, which, for example, Sanofi did. So we do have firewalls in place, and we haven't really had an issue with IP.
Ken has a second question. He wanted to know if there were any repeat customers beyond Sanofi who have agreed to presentation or publication, and he wanted to get some references for those.
Yes, so if you go to the Rosa & Co. website, under I believe it's under news, there's a whole list of posters that we have presented with clients. There are many others that have presented with us, and you'd be able to find that under the, you can download any of the posters that we've presented from the website. So it's actually too many to even mention, but at this point we've worked with most top 20 pharma companies, and we have publications with a good percentage of them.
Well, thank you very much. We're actually right at 10 o'clock now, and I want to thank you all for all your questions and thank Dr. Friedrichs for a very interesting presentation and discussion. And next slide, please.
Oh, yes.
One more slide. So I just want to let you all know that if you're interested in learning more about CPMS and our committee and the 10 rules that we're developing in our efforts, please visit our website. We also encourage you to continue the discussion about these rules, the rules that Dr. Friedrichs has presented, as well as our 10 simple rules on our discussion forum. And we'd appreciate it if you would take a few minutes to fill out the survey that's going to appear in a pop-up window at the conclusion of the webinar. And that will help us improve the webinars and choose upcoming topics. So thank you all very much.
And thank you.